we're going to get started. Welcome. Thank you all for joining us. We're really excited to have this opportunity today to have some interesting discussion and share some expertise. And we're looking forward to questions that folks have to share. So we are here today to dive into the exciting topic of biotech or biotechnology. So we have a number of different folks with us from a variety of fields and different aspects of biotech learning, research, professions. So we will get a chance to see how biotechnology has different impacts in the world and also different pathways to get involved in biotech that hopefully, might spark some of your interests. So we have a few folks from ASU here, myself included. I will be moderating this discussion today. I'm in the Mary Lou Fulton Teachers College at ASU. I'm also a former science teacher. We have a few folks from the ASU Biodesign Institute, so some of our lab leads here today, and on the science team for our project called Biosense Network, which you'll be hearing a little bit about today as well. We also have Dr. Stephanie Rogers from the Department of Defense, who is also involved in our Biosense Network as the funding body for our grant that allows us to do this cool work. And then we have really cool, we've got some students and also Ms. Rashna Nath, our teacher from Chandler Arizona College Prep High School, joining us today. So we're really excited to get the K-12 perspective in biotechnology as well, since that's a really, really important part of this pipeline. So I want to welcome all the panelists, and we'll give them a chance to introduce themselves before we get into our discussion and some audience questions after that. So here are our panelists today. Oops, sorry about that. So as you're listening today, if a question pops up, feel free to go ahead and type it in the Q&A. You can find this by looking at your bar, your Zoom bar that's probably towards the bottom or top of your screen, and look for the Q&A icon. Click on it and then go ahead and type in your question. I'll be monitoring that, and we'll be using those questions to direct to our panelists after our moderated discussion in the last 20 minutes or so of our event today. So please, as you think of them, before you forget them, you can go ahead and add them to the Q&A. All right. So we are going to go ahead and get started with our introduction. So each panelist is going to have a few minutes to introduce themselves and just kind of talk about what they do briefly. And so you can get a better idea of who all is in the room today. We're going to go ahead and start with Dr. Stephanie Rogers. So I'm going to pull up her slides here just a moment. And just a sec. Stephanie, go ahead and you can go ahead and start introducing yourself.
Great. Well, I'm Stephanie Rogers, and I'm very excited to be a part of this event and to be able to talk about biotechnology with all of you. I am at the Department of Defense, and I serve as in the role of Principal Director of Biotechnology, which in essence is I basically lead the department's strategy in biotechnology from both a technical and a strategic point of view, and work across all the different components of the department. When people think about the Department of Defense that aren't really involved in the Department of Defense, it's easy to think about the military personnel, but the reality is that there's a substantial part of the department that is non-active duty or non-military personnel. We're called civilians like myself, and we work behind the scenes to really support the research, the technologies, the strategies, the policies, everything that needs to go into actually enabling our military forces to do what they need to do. The Department of Defense came up with a set of technologies that are considered priority areas, and biotechnology is one of those. And this is exciting. I've been in the biotechnology field for a while before I was with the department, and so it's really exciting to have biotechnology named as a priority area, so we can really do a lot of incredibly important things for the department as well as the nation, and really accelerate innovation in this space. So it's really great to be able to support programs like this at ASU and others, so we can grow the expertise and the talent and the students to work in order to really capitalize on biotechnology. I did prepare just a few slides just to kind of introduce what we mean by biotechnology, as it maybe isn't quite clear just from the name of exactly why the Department of Defense may really care about biotech. So I just wanted to walk through a few of these with you as an introduction. Before I do that, though, I will say, just for my background, I've been with the Department of Defense for about 1.5 years now. Prior to that, I worked at a company called In-Q-Tel, which was a strategic investor on behalf of the intelligence community and the national security community, where I was leading investments in biotechnology. And I have a PhD in plant pathology, with an emphasis in forensic science before that. So, it's been, I've been involved in biotech for many years. Go to the next slide, please. Can you hit the next slide? I'm not seeing it change on mine.
Just scroll, I think.
Yep. Go hit the down arrow.
Hit the down arrow on the top.
It's showing it's advancing on my end, but I'm so I'm
must be frozen on my side. All right, well, we'll just
okay. Let me try getting out of full screen, and then
I can definitely talk about it.
Troubleshoot.
So while she's figuring it out, biotechnology, the way we define it is it's really an engineering discipline where we're leveraging biology, biological parts, or living systems to produce a wide range of technologies and products. So it's really leveraging what biology gives us in order to produce other things. Thank you. This is the slide I was looking for. And this can include things from food, cosmetics, medicines, textiles, fuels, a number of things that you wouldn't necessarily traditionally think are rooted in biology. Studies have shown that biotechnology will have a huge economic impact over the next 10 to 20 years, and will probably be one of the highest technology area contributing to economic growth. And so it's a very exciting time to be a part of biotechnology and grow this field into the future. The next slide, please. We've actually been making things with biology for a very long time. One of the probably most known things that's very public now is the production of things like beer or fermented foods. And so we've really been leveraging biology for quite some time. Really the way we do it is by using these tiny cellular factories, that's the yeast, and then we feed it food for it to eat, it's like sugar, and it's in water, and that converts that sugar and the ingredients that's in those big vats into the end product, like the beer, the wine, the yogurt, or other kind of fermented products like that. So go to the next slide. So this is actually what we're doing with the Department of Defense, but we're not brewing beer. we're producing things like fuels, and we're changing the input materials, the things that go into those big metal vats, to produce different things. And so we can, depending on what we put into those big metal vats, we can change what we're going to produce at the output, things like foods and medicines, fuels, all those things that I talked about previously. So the next slide. So just as an example of, I wanted to highlight 3 technologies that we're currently working on getting into production. And that first one is Biocement. This is actually using bacteria out in the soil and combining it with the nutrients that are in the soil, like salt water and calcium, and it actually grows the hardening material right there in the field. And it can create runways or roads, and it does this in a very sustainable way that doesn't require giant concrete machinery or a lot of heating or kind of difficult processes. And we can do this in remote locations. Mm-hmm. The second example is that the eye protection. So this is where we're leveraging biology to produce coatings or films, where based on what is produced in those metal vats, we can actually integrate that into eyewear, and it gives us the ability to tune or change the tint or the color of the eyewear to add some extra protection. The benefit of this is it can go beyond eyewear and actually can be used in LCD television screens, smartphones, smartwatches, or anything along those lines to help with performance and durability. And then the third example is medicine. So the COVID-19 pandemic showcased the need for novel advanced vaccines. And so the department has made major strides in the development of vaccine technology. And actually, the vaccine technology used for the COVID-19 vaccine was rooted in innovation for the department. There are other medical capabilities important to keep our active military protected. That includes environmental monitoring and personalized medicine as well. So that is a big area of importance. Next slide. So to develop biotechnology, as with any technology, there is a process. And so what you see here on the screen is a very simplified process, but just wanted to walk through these a little bit just to kind of showcase what it is that we do. We start with that earlier basic research where we're studying and understanding the specific questions in a specific area. That basic research is usually done at laboratories and with academic partners like ASU. And then we move forward into the advanced and applied research. And this is where we start to move into clear and practical applications, where you're looking into how something can be developed to be useful for a particular mission or operational use. This is also done often at laboratories and can involve academia or industry or both. Mm-hmm. The third state is the prototypes. And this is done once we're moving on to the stage of actually developing a testable item, like an engineered item, where we're developing it advanced enough to actually show what it could do in an operational environment, but only in testing purposes. And this often is done with some level of manufacturing. So you have to have maybe not those big, large vats that we saw in those pictures, but maybe something smaller or some kind of engineering prototype lab. So a little more sophisticated and can involve some of our defense industrial-based partners. And then manufacturing is the final stage where we're actually getting into the large scale production where the goods are really made. We're past the testing and now we just need to make it. The DoD labs that we have, as well as academia, do have some small scale manufacturing capacity that we can leverage, but there's also large players, the big companies that we work with within the industrial base. It can take years, sometimes even decades, to get through this entire process. And we've engaged with numerous labs across the country in order to do this. We did recently, the Department of Defense recently sponsored the stand-up of Biomade, our Bioindustrial Manufacturing Innovation Institute, to help partner with industry, academia, and other government agencies to kind of complete this, this pipeline here. I also just wanted to illuminate that the department does offer many different ways to engage along this process and to be involved in learning environments such as internships, fellowships, scholarships, other opportunities like that. The involvement of academia in this process is so important and it requires partnerships. So I encourage everyone to go to that dodstem.us link that's there, that website, and check it out. That's where you'll find all sorts of opportunities for scholarships and fellowships and other opportunities like that. So I just wanted to say I'm again very, very grateful to be able to be part of this. Hopefully that little tidbit is helpful, and I look forward to being a part of the Q&A as we move forward. Thank you.
Fantastic. Thank you so much, Dr. Rogers. Appreciate that. Yeah, and we'll get to dive in a little bit more. soon. So we're going to move on to Dr. Singaroy. If you could go ahead and introduce yourself in a few minutes.
Sure. Yep. So before that, I saw a number of questions came up. So will we be taking that at the very end, or can?
Yeah. So after we dive into some moderated discussion, we'll be able to address the questions that are in the Q&A.
Perfect. Okay. So hi, I'm Avi Sinha Roy. I'm an assistant professor in Arizona State University at the Biodesign Institute here. So we primarily work on, as Dr. Rogers was showing us, some of the early science and prototyping aspects of biotechnology. And so as a part of that, we work on a very specific part of it. We work on computational design. So when you actually go ahead and use a particular kind of technology, you have to conceive that. So a lot of that conceiving or the designing happens on a computer. And so can I share my screen quickly, Cassandra?
You should be able to, yes.
Yes. So some of that designing can actually be done by performing physics, chemistry, biology on a computer. And that's what we end up doing here in our lab at Arizona State University. It's called computational modeling. And so it is also something that, as part of this network, this BioSense network that we have come up with, and that Cassandra will talk a bit more about, that's what we end up doing. So we actually have students from different schools. We have students from ASU, undergraduate students, graduate students. All of us work together towards coming up with examples of biotechnology, but making models of them. So before really actually scaling things up in laboratories, even the smallest of laboratories, you really need to make sure that your technology is useful, and then there are certain rules that you can use to test the usefulness of technologies. Be it a biocement, be it a way of looking at light. Before scaling things, you really want to do initial testing, and it turns out computers are a very good way of doing this at low expenditure, so your return on investment is not terribly bad. And a lot of the work that we do at ASU in my lab is to do with modeling and computational testing of technologies. And we'll talk a bit more about that possibly as we move forward. But I think for introduction, let's say I'm someone who uses computers to learn about technology and then to kind of hopefully design new technologies. And we've come up with platforms of disseminating this way of modeling through BioSense network. And then we'll talk a bit more about that. So yeah, with that, I think we'll pass on to our next panelist.
Great. Thank you, Abhi. Okay. So we have Dr. Lapinet, who is also here at ASU. If you'd like to go ahead and introduce yourself.
Hi, everyone. Thank you for the invitation, and thank you for allowing me to share my my career path and my research in the lab. So currently, I am an assistant professor at ASU, and I'm also part of the Biodesign Institute. And before trying to explain what my lab is doing, I want to introduce myself and tell a little bit more where I come from and how did I get here. So I'm actually from Lithuania, and so that's a really small Eastern European country, and I did my undergraduate studies there and also undergraduate research, and it was very much basic research. And after finishing my undergraduate, I moved to Germany, where I did my PhD. And again, my graduate research was also very much basic research. So we were trying to understand how RNA protein complexes assemble and perform their functions. After my graduate school, I moved to Berkeley, where I had a chance to work with Jennifer Doudna, who most probably all of you know got a Nobel Prize in chemistry in 2020. And so in her lab, actually, this is where I started looking at how can we basic research, basic discovery, transform and translate into biomedical applications as well as biotechnological applications. And so my lab currently has really three directions. One direction is really very much basic research, trying to understand molecular mechanisms and pathways in neurons and neurodegenerative diseases. And then two other areas are more application or biotechnology, biomedicine related. So we are trying to understand bacterial systems and pathways in bacteria, especially how bacteria interact with their with viruses and with their pathogens. And understanding those pathways really allows us to discover new biotechnologically important tools and applications. And the third direction is really to try to translate those discoveries into actual applications. And specifically, we are working on CRISPR-Cas systems and genome editing and engineering. And even though my lab is really interested in how to use these systems to understand and treat neurodegenerative diseases, we envision that the discoveries and tools we will develop in my lab will be very important also for other applications, such as the food industry, agriculture, maybe even the climate change.
Fantastic. Thank you. So we're going to move on to Rashna Nath, our teacher over in Chandler.
Hello, good evening, everybody. And Cassandra and Avi, thank you for having me here. Dr. Rogers, it's amazing. Thank you for being here and helping us with this. And of course, Dr. Lapinet, thank you for being here too. Let me tell you a little bit about myself. I am a honeybee scientist. I'm a NASA ambassador, but I'm also a high school teacher and educator, which is my passion. I've been here for almost 6 years now. And through the 6 years, I have been able to establish a program which I call as DRIPPLE, which is Dream, Research, Innovate, Project-Based Learning or Problem-Based Learning. So the biggest idea behind this program is that a lot of students do project-based stuff, but what is the purpose behind it? The purpose is to motivate them to get to their careers or their dreams or their connect them with research. So I ended up doing DRIPPLE instead. So through that program, we have been extremely successful. My students, as early as 8th graders, when I started off, they have patent pendings and provisional patents in their names. And we have done a lot of projects that involves a wide variety of topics, starting from biosensors to, we have a prototype called Sense Hydro, which is a heat monitoring device, a temperature monitoring device for heat stroke. We are looking at a device that actually helps recyclables being identified by high school students in high school cafeterias. We look at styrofoam degradation using mealworms, scent bags that are VOC free because of allergens, which people cannot use, as along with air fresheners. We're looking at flavonoid band-aids. We're looking at flavonoid-based bioplastics, terraforming Mars, you name it. So whatever the students are interested in through the program, I facilitate that. And my collaboration with Dr. Shingaroy started in a similar concept where the basic idea for me is to be a facilitator of all of these amazing ideas that are in high school students. And I believe that if we catch these kids early in their research fields and we can give them a pathway, they will excel. And with all of your help, we definitely hope to get there. So I have 3 of my students here today. They're all my biotech students. I'll have them speak for themselves.
Fantastic. Thank you so much. All right. Yes, the stars of our show. No pressure, no pressure. Alex, if you wouldn't mind just introducing yourself, you can just kind of tell us, maybe what you're interested in school or what you've got going on.
All right. Hi, I am Alex Grumov. I am a student here at ACP High School, and I am interested in, I have 2 main projects. One is the Sense Hydro group that is developing that heat sensing device that users can wear and prevent themselves from getting heat exhaustion or heat illnesses. And the other is a project I've been doing since 9th grade, which involves kind of the neuroscience behind music and how music and the brain work together. And so that project kind of involved how music affected brain function. I investigated this through things like EEG analysis to a minor degree, and that's kind of my passion. biomedical engineering, but especially with how the brain works.
Cool. Super cool. Thanks, Alex. Caitlin, if you wouldn't mind introducing yourself.
Hello, I'm Caitlin. I'm a senior at Arizona College Prep and one of Mrs. Nath's biotechnology students. So I'm also working on the Sense Hydro project where we're developing that heat sensing device. And also outside of school, I've been working at a honeybee lab for the past 3 years. Let's see, in my sophomore year, I used honeybees as a model for caffeine addiction, and it was really cool looking at the neural networks in the honeybee and seeing how caffeine affected those. So I'm interested in neuroscience and also biology. And then this year, I'm focusing on studying active sensing in honeybees by using electroantenographies and seeing to what extent the honeybees use their antenna in searching for new stimuli and odors.
Awesome. Thank you, Caitlin. And we have Ahmad, if you wouldn't mind introducing yourself, please.
Hey, so I'm Ahmad. I'm a junior at ACP High School. Working under Ms. Nath has been amazing. So what I'm currently doing, I'm working in the biomedical engineering field with I've decellularized tissue. I'm making it into a tissue scaffold, and I'm able to apply that in a variety of ways, whether that be skin grafting for burn victims or just basic bandages. I've been able to work under like the biotech industry, especially with all the resources that we have here. So I guess really my interests, they kind of just revolve around the biomedical engineering phase of all of this, and especially around the biology aspect and how we're able to harness, modify, and engineer that entire shebang. That's kind of my niche.
Great. Fantastic. Thank you, everyone, for introducing yourself. So I do have a few questions that we'll go through. Some are specific to certain folks and some are just open for anyone who wants to answer them. So I'm going to get started. This one is for Dr. Rogers and Dr. Lapinet. How did you get to be where you are today? So I know you started talking a little bit about that, but are there any sort of highlights along your journey or maybe barriers along your journey that you could tell us about? Did you always know you were gonna, be in the sciences, or what path did your life take to get you to where you are now? So either of you that wants to start off.
Happy to jump in. So I love this question because the big answer is that if you told me 10 years ago that I would be at the DoD or any position like this, I would have said, ain't no way. It was not something that I visioned. It wasn't anything that I set a path for, but I knew I wanted to be in sciences, and I love learning about science and then just applying it to some item of need, something that can help the people, help society. And so I thought I was originally going to be a DNA analyst at the FBI. That was my career track. That's where I wanted to go. And I was all set to go there, get a master's degree to do that. But then I learned about the importance of plants and the health of our plants and learned about a program. And so then I ended up shifting kind of spontaneously to chase after that because it just felt like a really important need for society. So I wanted to go that way. And then I just kind of went where it took me. And honestly, it was more opportunities came available. So the biggest thing I can say to those that are up and coming is leave doors open. Even if it seems crazy, say yes, because you never know where it's going to take you, and find your passion and know kind of what keeps you going and what allows you to thrive, and jump into it. And so that's basically what I've done. That's my philosophies throughout my career, and it's landed me here, and I couldn't be happier. So
fantastic. Thank you.
In my case, most probably it's a little bit different story because I always knew that I wanted to be a scientist since most probably since I started my undergraduate. And the more I studied, the more I had experience in the lab, the more I knew that that's definitely what I wanted to do. I did not know, or I did not, in the very beginning, I did not think that I will be a professor at the university or that I will have my own lab. I just knew that I will do research. I also knew that I wanted to stay in academia in some way, because I thought that the great part of academia is that you can work on your own ideas, of course, if you can sell them and get funding. But in a way, it's your ideas, the things you care about, you can really push forward and not necessarily depend on, let's say, a vision of a company. So I thought having this freedom in academia was something I was very excited about. During my graduate school, so basically most probably second half of my graduate school, this is when I started carefully dreaming about that one day maybe I will have my own lab. And definitely after I joined the lab of Jennifer Doudna after graduate school, I knew that this is what I want to do, just because meeting great scientists who really changed the world through basic research, that was something I also wanted to do, and I hope that my lab will contribute to the development of new biomedical applications as well as biotechnological applications. The most important thing, which I think, at least for me, there were two things which I think helped me to be where I am now. One is really have supportive mentors and advisors who really cared about my success, and just running some ideas by them, talking to them, and getting their feedback, I think was really key. And the second very important thing is really to have a supportive family, even though they may not be in the field, they may not understand what you are doing or what you want to do, but just have to have people on your side who believe you and who support you from just everyday routine stuff, right? And really helps you to invest your time into your career and your research. I think that was also very, very important.
Great. Thanks so much. So this next question is for our ACP High School crew. What do you love about teaching and learning biology? What gives it juice? So that's for Rachna, for any of the students, whoever wants to chime in.
Oh, I'll go first. I mean, what's the juice? You can see three of my students sitting here talking to panelists. The juice is basically to see them shine bright and do what they truly love to do and get the maximum out of it. And that's the reason why I do what I do, just connect them.
All right, I guess I will go next. So the juice of what I do is, or I guess the juice to it, or my motivation is just being able to learn something new. At the heart of it, I am a very curious person and I want to know, I want to understand everything. And something that I found is really interesting to me is the way we think and the way we act. And I really enjoy the scientific aspect of neuroscience. So that's kind of why I chose that sort of pathway. But you'll see that I like to engage in like a variety of projects because I want to expand my knowledge in kind of all of science and technology and just however I can.
Cool. Caitlin or Ahmad?
Yeah, I think what I love about science is just the hands-on aspect of it and being able to formulate your own questions and actually carry out experience in different tests to kind of like get evidence and support for your own questions and answer those questions for yourself. And along the way, I'm always like learning something new about science and new techniques. And I think it's really cool how technology and science have come together. And there's so many cool different like aspects and applications to it. And that's just like what I love about science. It's like a never-ending pit of knowledge.
Yeah, so building on that, like especially being a little younger and then, wondering how wondering about how everything like revolves around each other, works with each other, the motion with each other, right? And then being able to really like focus into the science of that, how our body works, how our body reacts to stimuli. A lot of that has been very fascinating to me. And honestly, it just kind of builds your understanding of the world in itself. Building more on the biology aspect and branching out into the branches that actually are offered, especially at ACP, or in the world, like academia in general. Engineering with biology, it kind of brings a really new aspect in how you're able to harness biology, how you're able to harness not only your surroundings, but also yourself, and make yourself either better adapted to this world that is everlastingly changing, or, it's just really about communication with yourself and also the outside. That's probably my juice.
Cool. Thanks. I love that idea of you becoming, immersed in the science and you being part of it. That's a cool idea. So my next question is for Abhi, Dr. Singroy. What is the most exciting thing about what you do? And how will you share that excitement through this Biosense Network project that we've talked a little bit about?
So yeah, I think again, again, just to go back to from where I come from, like my training has been, in a way very similar to Audrey's, like coming from a different country, learning maths, learning science, learning art and literature, and then having to make a choice, whether to move forward with science or to move forward with art and literature, and then to choose science, then to see where science takes me. And the thing that excites me the most is, of course, I'm trained in chemistry, so that's my fundamental training, and then I moved on to physics and biology. The one thing that often intrigues me is how small of a thing can I look at, like how small can you see things? How can you make invisible things visible before you? So essentially, you can take a guess, like, might be you take a piece of hair, that's the smallest thing that you can look at. You shade it even smaller, you can't end up looking at those things. So how can you come up with microscopes to look into objects that are very, very small? That is something that I feel to be extremely exciting. And I found that there are certain ways in which of course you understand when you see things, you have to understand how light is interacting with something. And so how can you see essentially how can you gauge how light is functioning is what excites me the most. And so I wanted to make microscopes. Well, it turns out I realized soon enough that making microscopes is not very easy to do. And you need to learn a lot of things to do that. And of course, life is finite. And as myself, as one person, it's not so simple to make a microscope. Or if you work in a much bigger group, and which you do often, you end up making these big microscopes. And in fact, famous scientist Feynman said that most of the science is done up to 1960s. After this, what we're going to do is make good microscopes. So then I said, okay, fine. There's so many people who are doing microscopes, really making microscopes. Can I start using a computer as a microscope? And so then that's where essentially this idea of using computational microscopy comes in. Like, how can you start seeing the smallest possible objects in 3 dimensions that you cannot see otherwise? Either you need these huge microscopes, which are very expensive, or you can like have something on a computer, build a microscope on a computer and kind of see it yourself. And then you make sure that you come up with ideas that everyone can see using that microscope. So that's what I think the most exciting thing is, the preparation of that computational microscope. And then I think in the context of Biosense Network, where we are bringing this microscope, we are now giving this microscope to the students and to high schools and to middle schools, so that they can see it for themselves, how these molecules that they have been seeing in their textbooks look like in as real of a situation as they can be. So yeah, I think, and of course, the connection to biotechnology is essentially a technology becomes a technology because certain molecules, their properties can be exploited in a particular way. And so you really need to see, often we see technologies in their manifestation, which is how things look at the level of forensic sciences, at the level of plant sciences, at the level of our usage. But things that is beneath that is very, very interesting to look at. And so this computational microscope allows them to look into things as complicated as Audrey was talking about, CRISPR-Cas9 kind of a system, or artificial photosynthesis, or vaccine design. How do they work? How are they technologies? What are they exploiting? So to look into the molecular principles of those using a computer microscope. And that's what our students will do as part of this Biosense Network, where we have now placed our microscope on the internet. So essentially there is a web browser. So let's call this software called Computational Microscope is now available through any standard web browser. And so students can actually access that and then start looking into the molecules of their interest, first in their textbook, and then they complement that with this three-dimensional visualization and interactive visualization where they can play with it. We have computational tweezers where they can use to tease these things apart. And yeah, and so that's what that's where my enthusiasm of seeing the small things and the molecular life overlays with this idea of conveying technology through these molecules.
Great, thanks. Are there any of those the content modules that we're developing through that project that you wanted to highlight to give folks an idea of what students and teachers will be engaging in?
Sure. So we made a small movie of that. After I've shown the movie, you'll have to tell me who's the voiceover on that movie. Okay. So that would be it's a celebrity. I can tell you that. Okay. So let's let me share the screen. So as I said, a molecular. So here is, you guys can see my screen, right? So essentially, this is a journey through a cell. This is what life at the molecular level looks like. The movie that I'm going to show has nothing to do with technology. Technology definitely, basically exploits such molecular details. So let me first just show you this. So this is a movie that was, is it over? Oh, sorry, it's, I went to the last frame. Let me go, start from here. So let's go a little bit behind here. Okay, let's have a look at this movie. This is what a computational microscope is going to show you. And I promise you by module number 5, we'll be able to see something like this. Every student will see something like this on the computer. But let's have a movie.
Photosynthesis, the process that fueled life, made its first appearance in simple bacteria over 1 billion years ago. Using one of the world's most powerful supercomputers, scientists have sought to model its chemical secrets. Step 1 is to capture sunlight. Passing through a transparent membrane to the center of the bacteria, we encounter a cluster of spherical pods. Each one is equipped with special sensors designed to harvest solar energy. When a photon hits, it excites rings of chlorophyll molecules.
So should I stop it, Cassandra? Because might be in the interest of time, I think we should, or...
This energy is transferred across a family of proteins.
So let me just...
the machines of life.
Accelerate this.
Down the line, electrons.
So essentially, yeah. So this is like kind of a view of life in its gory details. And a lot of biology that you see here end up utilized, leveraged in biotechnology. And that's what we will try to show our students. But again, this is a view of a bacteria. Like you cannot see a bacteria with your, with the normal eyes. You need a microscope. And that's what we end up, doing. So this is actually module 5. And in module 5, people will actually interact with an object like this. So yeah, I think that's what I'm very, very excited about in terms of conveying the molecular view of life, which normally if you see in a textbook, you just see some cartoons and nothing more. But here is the reality, as real as it gets. So yeah. So whose voiceover was that? Can anyone guess?
Got me.
Okay. Sorry?
Got me? I don't know.
Yeah, so I think, no, so originally it was supposed to be Benedict Cumberbatch, but he did not. So we finally got Jon Snow from Game of Thrones. I think it's not for the kids, but when you grow up, you will see. So
I don't think that's his actual name, but yeah.
Yeah. So the actor who played Jon Snow actually did voiceover for this movie, which was actually the best visualization showcase in 2020 in one of the conferences called the GPU conferences. But again, this computational microscope runs on very special hardware called the graphical processor units that ASU has. That's the reason we can do this. ASU has some of the biggest computers which we can leverage, and then convey this microscope to everyone. So yeah.
Cool. Thanks, Avi. So this question is open to anybody, kind of focusing on the technology part of biotechnology. How can biotechnology make the world a better place? Sort of focusing on the so what, why pursue this? Why does this matter for humanity, for ecosystems, for the world? How can it save the world? How can it make the world a better place?
I'm happy to jump in, although
Yes.
others probably have an answer too.
I think you should start.
Well, when we think about, there's a number of challenges that the world has. I mean, not everybody has access to food. Not everybody has access to healthcare. We have this thing called climate change that we're dealing with. There's a lot of challenges, and biotechnology has a solution for basically all of it. It's just a matter of harnessing those solutions and actually getting them to reality. The reason we're able to actually move into the application space of biotechnology is because of all of the amazing basic research that's been done in biology to understand it. I mean, that movie that we just saw is incredible. Like, I didn't have that when I was in college, but to be able to actually understand biology and its fine-tuned details, it allows us to actually capitalize on it and start to engineer it and design the things that we need to actually get to real solutions. We can start to grow food in climates that we never were able to grow them in before. We can clean water more rapidly and effectively, and even use portable systems to do that when we're hiking or for the military or whatever purpose it may be. So it's just we're now at a place where we can actually wrap our hands around biology and actually use the knowledge of biology to produce solutions. And I've always been a fan of nature, because nature is the ultimate engineer. I mean, when you think about the diversity of the organisms that are out there, we have barely even scratched the surface to really even understand the complexity and beauty that nature has given us. So I'll turn it over to the other panelists for additional input.
Thank you, Dr. Rogers.
I think Audrey should go next.
Well, I totally agree with Stephanie that nature is the most creative and the most amazing thing we have, really optimized through millions and millions of years, right, to address some of the most important challenges we face today. And I think we definitely learning from nature, learning from basic research will make our society happier, healthier. And I think the one thing which we need to think more and more about is that it's great that we have already so many biotechnological solutions, but how do we make all of these solutions really accessible to everyone, independent on geography, independent of economic situation, and really that even talking about biomedical solutions, right now, many of the most advanced technologies are available only to the very few people who can afford that. So how do we make this really much more affordable and more accessible to everyone? And I'm of course talking a lot about this from perspective of genome editing, because of course everyone is very excited about being able to treat rare diseases, to be able to treat diseases we were never able to treat before. But how do we make sure that everyone has access to technology?
Mm-hmm. Might be Rachna can say something, and then I will.
Well, when I talk about biotechnology, I see prospects, I see avenues, I see dreams coming true for students as young as 9th and 10th graders, because unless we expose them to these technologies, they have no idea that it exists there. And that is what my goal is, again, as an educator, to see that these students get exposed to the maximum capacities beyond even their wildest imagination, so that every time they see in a microscope, they're like, wow, what is that? Instead of, oh, I don't know what that is. So that fascination aspect, the more we cultivate in them, I think we're going to get more and more better results in the future, because curiosity is the basis of invention. That's what I think so.
Yeah, and I can say that, like biotechnology, it saves lives. If you see, like during COVID, for example, like there is so much that happened to, like, we got the vaccine so quickly. Why, how did that happen? So essentially, I think biotechnology has its, I think here is my thought about, like, if I have to compare this biotechnology and there is, let's say, non-biological technology, right? Let's put it so. I think biology or life is not the most efficient. We are examples of biotechnology. We are kind of defective in our own ways, right? Everyone have their quirks, everyone have their rights and their wrongs. But what we are is we're very adaptable. Biology teaches us to be adaptable, to kind of exist in a lot of different situations. We might not be accurate, but we are we're very adaptable. It's the scientific word for that is, I'm forgetting the word. It's called robustness. We're very robust. In comparison, normal technology, materials technology, is very accurate, very precise, but you can't just do it all around the world. You have to have a very particular kind of setting, very clear instructions. Then it goes into a black box and comes output. So I like biotechnology for that adaptivity, for that, for the fact that there's a lot of errors that it actually absorbs within itself and still give you what you need. Might not be in the most efficient way, but in a way such that it can sustain you for, let's say, a lifetime of 80 years on an average. So I really like that I would say advantage of biotechnology over traditional non-abiological technologies, if I may, where I can create a technology that is very robust, very adaptable, might not be as efficient. That balance of efficiency and robustness really attracts me towards learning more biotechnology. And yeah, I think.
Great. Any other thoughts? Any thoughts from our students about, the world that you're becoming adults into, and, what the role of biotechnology is in that world, why that's important?
Yeah, so as the new generation that's kind of upcoming, it's almost as though we're inheriting a specific type of world that we need to be able to advance biotechnology. For example, with COVID-19, we needed to quickly and efficiently develop a new mRNA vaccine. So that required a lot of research. As we get into more specific diseases, I think that there will be a lot more emphasis on more molecular solutions. And as technology gets more precise, I can definitely see kind of new fields and new research opportunities in more precise fields. So I definitely see an importance in that and opportunity in that sort of aspect.
Building on what Alex was saying, when you kind of ask, so what, why are we using biotechnology? And especially when we're talking about our generation, me, Alex, and Caitlin, we're starting to see some sort of trends that are ushering in more and more solutions. And, as, no offense, as you guys are on your exit, we're taking entrance and we're starting to, take control of our environment, the things around us, and we're trying to find solutions for that, right? So with biotechnology, it's no longer we're trying to solve it at the surface level. We're going into the semantics, we're digging deep at the actual root of it, and we're using biotechnology to solve those problems at the root, at the start. We're essentially perfecting humans' adaptability, and again, as Dr. Abi said, our robustness. So that's more or less the impact that biotechnology has for us.
Yeah, I also agree. Biotechnology is so versatile. We can use it in so many applications. And yes, we're going back to that molecular level. We're also going to see new advancements, but I think it's also important to make it more feasible and affordable to everyone, because what good can it do if no one can use it and afford it for themselves?
Definitely. Awesome. Thanks for all your thoughtful, insightful answers, everybody. I would like to invite attendees, if you haven't already, I know there's been a number of answered questions in the Q&A panel right now, but if you have a question you'd like to direct generally or to any specific panelist, I invite you to go ahead and type that in the Q&A box right now, and I'll keep an eye on that. I do have some other questions while we're waiting to see if folks add some questions there. So far, I've heard things like curiosity, open-mindedness. What are some traits or habits of being that, in your opinion, are essential in biotechnology, pursuing biotechnology, or just in the sciences in general? Ways of being or mindsets or traits.
I think definitely persistence. Don't give up if it doesn't work, because science usually doesn't work most of the time, and experiments fail and fail and fail. And so just make sure that with each failure, each failed experiment, you learn something new, and just be persistent and go after what you want to know, go after what you need. And I think that's the most important. And I think it's not just for science in general, most probably.
Yeah.
I would say troubleshooting. So don't be afraid of breaking things, but like once you've broken it, learn how to fix it. That process is going to teach you a lot of things. So again, as kind of Audrey said, it's for everything, but specifically for science, because persistence persistence is a general characteristic. Like you have to be persistent to live. Troubleshooting, you can is something that very specifically helps you. So I think learn to break things. Don't be afraid of breaking things, but have the persistence to make it back, to fix it. That's very important.
I would agree. Persistence is very important. And to kind of what you were saying, I would say be willing to be a risk taker a little bit, but of course within the bounds of ethics. but because that when we're not afraid to try something that we think may be a good idea, that's when we can really kind of stumble into some really fascinating stuff. So I would add that. And then also, I think science is inherently interdisciplinary. So while it is really important to be kind of deep in your own area, it's also so important to be communicating with other specialists, technology experts, people outside of your normal wheelhouse. And that's when we can get some really interesting discoveries going.
Yeah, here at the Teachers College in ASU, we one of our kind of foundational lenses is principled innovation. So not just move fast and break things, but go slow and nurture things.
Just to add on Stephanie's comment, it's also, the time when you alone as a single scientist can, change the world is in a way over, right? All the biggest discoveries and the biggest impact is really done by teams of scientists. And I think it's very, very important to be not just, able to work on your own ideas, but also to find people who have similar ideas or maybe similar want to try and solve similar problems and really try something find in common and because you're empowered by your colleagues and other scientists who have maybe different perspective on the same issue.
Exactly. And I would quickly add, ask why. Very important. Every time, whenever you get a piece of information, ask why. The reason I came up with Biosense Network, I'm a father of a 6-year-old daughter. So my kid, keeps asking me why. And I really felt like, she was looking into reading these books and can I have something? That's very boring for her. If I want to teach her something that is interesting to her, it's very difficult to kind of convey that. It's very cartoonish. So can I, on a computer might be, she's not very enthusiastic and interested and she wants to see things. But I think asking why is very, very important. So that will give you the questions. And then of course you need all the persistence, all the ethics, all the troubleshooting. But asking why is, first of all, often people have the answers, people don't have questions. So if you come up with a good question, then I think you're doing good. So, yeah.
Great. I do want to just pose this question to Dr. Rogers. Do you know anything about how the Department of Agriculture is finding ways to use biotechnology or anything about biotech applications in agriculture? I know you had mentioned, food insecurity being a big issue in the world. Anything you say about that?
I can't speak on behalf of USDA, but I do know that biotech is a very important area for them as well. In particular, food and feedstocks for manufacturing or other processes. Agriculture includes animal wellness and medicines for agricultural animals. So all of those ways that we think about, that I think about biotech, they have a similar view as well, and they have a lot of efforts underway themselves to make their food supply and agriculture systems more resilient. So it's definitely a big focus for them, but I'd have to defer to them to give details.
Great. Thank you. And I did see this question for Ahmad. What advice would you give to your 14-year-old self? That could also apply to others too. Any advice you'd give?
So honestly, I would give my 14-year-old self a smack because I really lacked the ambition for, I lacked the ambition for, biotechnology. And I won't lie, a lot of my ambition did come from Miss Nath. It's, so I didn't necessarily like take in or immerse myself in like the opportunities that I was presented to until, Miss Nath was like, because she taught me biology in 6th grade. There's a long pipeline of us, but she's like, oh my God, how's your project? How's this, how's that? Here's my extending hand. I run a program. It's called Honor Science Research. I joined that, and ever since then I've been taking it off. So the advice that I would really give to my 14-year-old self is that if you have an idea, there's no reason at all why you should doubt yourself. You should always just stick to it. If you have any idea, just go for it. Go for it. Unless it's horrible, but
Awesome. Well, with those very wise words, we are going to close out our questions. If we receive any others in the next minute or so, our panelists will do our best to just type an answer real quickly. But I do want to thank everybody. Thank you to each of our panelists for taking the time to share your world with us. And thank you very much to all of our attendees as well for coming here and listening and sharing your questions and your interests in our panel today. I will put up the contact information here for folks if you are interested in what you've heard today and would like to follow up with some of our panelists today. Here is their contact information. And I'll leave that up for a minute, but thank you all for joining, and hopefully we satisfied your curiosity today, maybe give you a little inspiration, and we appreciate your time. So have a great rest of your Tuesday.
Thank you all.
Thank you, everyone. It was really a pleasure.
It was.
Absolutely.
Bye.